The two most common type of bacterial structures or morphs are going to be coccus and rod. This is true for both the eubacteria found in the domain bacteria and the archaeobacteria found in the domain archaea. But there are other shapes as well. So some bacteria will have a Vibrio shape. Vibrio is kind of like a curved rod. Some bacteria are going to be spiral shaped, and we have two different types of spiral shaped bacteria. We have spirillum and then the spirochete. Spirochete's kind of like a really tight, if you remember the different types of perms, this would be like a super tight curl, whereas spirillum is more of a loose curl. And there are some species that actually can have different shapes. Typically, the organisms that have more than one morph are going to be found in the domain archaea, but there are some few bacteria that do have more than one morph, and that is termed pleomorphic. When bacteria divide via binary fission, sometimes they stick together. So that doesn't mean that they're forming a multicellular organism. They're just forming a multicellular arrangement. And so when bacteria divide, if they're a single coccus cell, they can divide and stick together, forming diplococcus, or a chain of cells called streptococcus. If they're a single coccus shape, they can also form square or cube-like packets. This is called sarcina, or clusters, that's called staphylococcus. The same is true for your bacillus. Bacillus cells oftentimes will stick together, forming streptobacilli. I just don't have that picture here.
Prokaryotic microorganisms exist in a variety of different cell shapes. Some are spherical, but seldom are seen as a single cell. A spherical cell is called a coccus. Pairs of spherical cells are called diplococcus, the plural being diplococci. Chains of cells are streptococci, and grape-like clusters of cells are staphylococci. Some genera, such as Streptococcus and Staphylococcus, derive their name from the most common pattern of arrangement of the cells. Many prokaryotes are rod-shaped in appearance. A rod-shaped cell is called a bacillus. The genus Escherichia consists of rod-shaped cells. The genera Bacillus and Clostridium consist of spore-forming rods. Spore formation in some bacteria causes swelling of the mother cell. There are also many other genera of rod-shaped prokaryotes. Bacilli occasionally occur as chains of cells called streptobacilli. Cells of the Corynebacterium group have a characteristic club-shaped appearance. Some bacteria have a curved rod or vibrioid appearance. An example is the genus Vibrio. Mycoplasmas are a group of bacteria that lack a cell wall. The cells of this group are pleomorphic, meaning that the cells take on a variety of different shapes. There are two groups of spiral-shaped bacteria. Spirilla are motile by means of flagella, and spirochetes are motile by means of axial fibrils located beneath a sheath that covers the entire surface of the cell. Bacteria such as the nocardiae form branched filamentous cells. These cells reproduce by fragmentation. Actinomycetes form branched filamentous cells. This group is characterized by formation of a variety of specialized dispersal cells that form on the aerial mycelia. In some species, these cells consist of strings of individual small cells, whereas in other species, the dispersal cells are found in sacs called sporangia.
So what we're going to do now is we're going to look at the different structures of the prokaryotic cell, including the plasma membrane, the cell wall, the nucleoid region, the structures associated with the cell that allow the bacteria to either move or stick to surfaces. The first thing we'll look at then is the plasma membrane or the cytoplasmic membrane. This is a phospholipid bilayer just like in eukaryotic cells. So you can see that phospholipid bilayer here. It contains two layers of phospholipids. The heads are always associated with the extracellular environments where there's going to be fluid because the heads are hydrophilic, which means they are water-loving. The tails are hydrophobic, water-hating, and so they stick together kind of like the meat in a sandwich. So you can look at the heads as the bread and then the tails as the meat. The plasma membrane is a semipermeable membrane, and it is basically a barrier for allowing certain materials to move through. There are a lot of proteins associated with the plasma membrane that will allow for other functions as well. The cytoplasmic membrane is selectively permeable. This means it will allow certain molecules to cross directly through while holding other molecules out. So it's very picky about what it allows in or out of the cell. Small nonpolar molecules are going to be able to move directly across the plasma membrane through a process known as diffusion, simple diffusion. Larger molecules or polar molecules like ions will have to use special transporters. And then if they are really large or if they're moving against a concentration gradient, then we're going to be using active transport mechanisms. I will mention that water, you can see water here, can move directly across the plasma membrane through simple diffusion, but typically water will move through a protein channel called an aquaporin. Water is a polar molecule, so it's a hydrophilic molecule. Water-loving, water loves water, but it's a special molecule. So it can pass through our cells, it can pass through bacterial cells readily. Simple diffusion then is the movement of molecules from higher concentration to lower concentration down a concentration gradient. And molecules that are small or nonpolar are going to move across the plasma membrane via simple diffusion. The diffusion of water across the plasma membrane is called osmosis. There are three types of osmotic solutions. Hypertonic solutions have a very high concentration of solute in the solution, and then the cells that are in the solution have a lower concentration of solute. So they have more water inside of them. Isotonic solutions have equal amounts of solution water in the solution as is in the cell. In hypotonic solutions, we have more solute inside the cell than in the solution surrounding the cell. So with hypertonic solutions, since there's more water inside the cell, water is going to move from higher to lower concentrations out of the cell, causing the cell to shrivel up a lot of times. In hypotonic solutions, since there's more solute inside the cell than outside, water is going to move into the cell and cause the cell to get very large and potentially burst. But with bacteria, most bacterial cells have a cell wall. In fact, I would say there are examples, Mycoplasma pneumoniae. Yeah, Mycoplasma pneumoniae is an example of bacteria that does not contain a cell wall. But in general, they all have a cell wall.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
The last thing I'll mention in this lecture here is that sometimes molecules have to pass through proteins. So we have protein channels and protein carriers that will move molecules across. This movement of molecules across can be in a passive mechanism like facilitative diffusion or in active transport where we're using some form of energy.